It's an honor, of course, to talk about it in William Withering's name. I only discovered this morning that the talk was somehow connected to psychotherapy, and I said this to my wife, who is a psychologist, and she said, that's absolutely adequate, because if you know what psychotherapy is, and I had to admit I didn't, it's understanding the nature of your obsession. And so I have had EBV as something of an obsession for some time, since I joined Tony Epstein's lab in 1872 or 1972? One of those 2 years. It starts with Dennis Burkitt, who was Withering-esque in the breadth of his vision and the breadth of his interest. He's most famous, I think, among the general public for his work on fiber. But his work identifying Burkitt lymphoma was a classic of clinical observation. He recognized this as a tumor which he had never seen before. He mapped its geographic incidence around Africa and instigated a huge area of not just cancer epidemiology, but oncogenesis in general. Because when we look at this tumor, which is a monomorphic B-cell lymphoma interspersed with macrophages, which turned out eventually to be eating apoptotic cells, he didn't realize the significance of what he had seen. Because if we just look at this as the archetypal human tumor, the Rosetta Stone, which has opened so much in oncology, it was the first chemosensitive human tumor. He treated it with cyclohexamide, the only drug at the time, and he said he was the worst chemotherapist in the world, getting the best results. And that was because this tumor, as we'll see, is highly apoptosis prone, and hitting it with a cytostatic agent drives the apoptotic program. Of course, it's the first virus-associated human tumor, and of course, it was also the first tumor linking chromosomal change to a specific oncogene activation. So these amazing triad of properties which makes Burkitt such a model for our understanding of the process of lymphomagenesis. Now, Tony Epstein met Dennis Burkitt in 1961 in the Middlesex Hospital when Dennis was giving a seminar about his work in Africa and about this unusual lymphoma. And after 3 years of frustratingly searching in biopsy cells for this mythical virus, which Tony hoped would be a arthropod-borne virus, which was restricted to areas in which Burkitt's was endemic, he finally cultured the cells. And how lucky that this is one of the human tumors which can be cultured, and how lucky it was that he had the electron microscope, because the virus that was there was not sensitive to detection in any of the contemporary virus search assays that were available at the time. And it's an interesting thought experiment to think who would have discovered it if it hadn't come this way. And I've got some thoughts on that, but I'll share you with them in the bar. Here is one of the pictures that Tony took in those days, almost exactly 50 years ago now, and here's the first paper published in March 1964. Obviously a herpes virus particles, but as it soon turned out, it was a herpes virus that was widespread in the human population. So there was extreme skepticism about the significance of this association. An agent only seen by electron microscopy in cells cultured from a human tumor, but an agent that turned out to be infecting every population on the planet. Now, the heroes of the field, in my view, after that initial discovery are here. They're both husband and wife teams. Werner and Birgitta Henley, working in Philadelphia, had a golden age in which they established the seroepidemiological tests for the virus, which actually we mainly still use today. They showed that virus can transform lymphocytes into permanent cell lines, and fortuitously stumbled upon, when one of their technicians developed infectious mononucleosis, and happened to turn from their seronegative control in the lab to a raging seropositive, that the virus also caused infectious mononucleosis. This happened in a couple of years, and it was while Volker Diehl and Harald Zuhausen, who went on to win the Nobel Prize for his papillomavirus work, were working in Werner's lab. And both of them were at this meeting in Oxford 1 week ago, talking about those days with great affection. The other heroes are George and Eva Klein. George is an inspirational leader in the field, intellectual powerhouse, who helped to develop the assays in seroepidemiology, promoted work in his lab, which led to the identification of the chromosomal translocation, where the MIC-IG fusion gene was cloned, and with Beverly Reedman, a young postdoctoral fellow coming for a year to the Karolinska in 1973 from Brisbane, worked out the staining to find the first marker of latent infection, the so-called Ebner protein, which was the only Ebner marker of viral infection that we had for almost a decade. Now, Tony, I learned a lot from, in the sense that he was a very clear thinker. I used to go along, I remember, with this idea or that, and he'd say, just stop it, let's just focus on the main event. And he actually was talking to me then in early 70s about vaccination. And he was always talking to me about the possibility that the post-transplant lymphoproliferative diseases that were just then being recognized in long-term kidney transplant recipients might be EBV-driven. And I just thought this was just too much to imagine, and just show me this evidence, show me that, at least I'm sure this is right. He was also a great stickler he's a grammarian, and he would go through your papers with a tooth comb looking for syntactical errors, grammatical errors, and so it turned out, it never stops actually. Here he was about 50 years later, talking to Dorothy Crawford at this meeting last week, and he's just saying, well, sorry, Dorothy, but you still haven't quite got it right. So that was the background. Of course, let's think about EBV, and we've been thinking about it as a 50-year virus, a bit like we've been thinking about Birmingham as a 50-year virological experience. But that's a kind of anthropocentric view, isn't it? I mean, the reality is that, of course, it's been around for millennia. And I got this from Duncan McGeoch, and you can see here are the herpes viruses arranged into their alpha, beta, and gamma subgroups. And we're dealing with this particular subgroup, which are the gamma-1 viruses, or the lymphocrypta viruses. And you'll see 3 archetypal members here, but we'll just look at this slightly more detail, just to give a kind of long view to what this virus can tell us. So here is a evolutionary diagram of the speciation of Old World primates. This is human. Actually, it's Fred Wang, my friend in Harvard. And the rhesus monkey at the other end, and all the way through these Old World primates and monkeys and the higher apes, one can see EBV cousins. And so clearly the virus had evolved through speciation of the Old World branch of primate evolution. For many years we imagined, because there was no serologic cross-reactivity between Old World and New World animals. New World animals did not have antibodies that cross-reacted with EBV proteins, that there was no equivalent within the New World primates. That all changed, actually. Oh, let's just look for a moment at the degree of homology between the Rhesus virus and EBV. And you see that it's very strong, exactly the same genes in the same gene order, some loss of homology, but particularly in the latent genes. So let me introduce you to the genes you've already heard a little bit about today. EBNA1, the other members of the EBNA family, and the 2 LNPs. And if you look up here, you'll see that this is where homology is becoming weaker between the rhesus and between EBV. So as the viruses have speciated, diverged with their host species during Old World primate evolution, what has really been driven is diversification of the latent genes. We still don't really understand what this means, except that these viruses have got to learn to live within the B-cell system of different hosts. They have to learn to interact with the physiology of the B-cell system in those different hosts. And it's the latent genes that do that, whereas the lytic genes, the ones that are well conserved, are largely replication machines that work in any cell type if you really drove it. Now let's look at the new worlds, because in 2000, something remarkable happened. A marmoset colony in one of the primate centers in the US developed a severe lymphoma, from which Fred was able to isolate a virus which looked for all the world like a cousin of EBV, not a close cousin, but a distant cousin. And we now know that many of the New World primates, you can amplify from their blood, you can amplify sequences which are clearly EBV-related sequences. So the gamma-1 herpes virus is stretched to distant cousins now. And if you look at the marmoset virus now, which has been sequenced by Fred, overall homology drops below 50%. So even the lytic genes, the replication machinery genes, are distinct, and that's why you don't get serologic cross-reactivity. And also look at the latent genes now. The EBNA1 is there, but it's a very distant, pale image of an EBNA1 as we understand it. The EBNA3 genes, which are highly related and have evolved by duplication, are actually down to a single EBNA3 protein, EBNA3 gene here. EBNA2 is there, but is very distant, and the LMPs are there, but they are so distant that we can only be recognized positionally. They have essentially no homology. And in fact, this LMP2 gene looks like a KSHV protein, looks like one of the KSHV latent proteins. So just a hint now as to how the gamma-2 viruses, which preceded gamma-1, might have given rise to a primate, a B-lymphotropic gamma-1 virus. Okay, so what can we learn from this? These are ancient viruses, they're not just 50 years old. They replicate in pharyngeal epithelium, and they essentially form latency, an antigenic silent latency, a bit like simplex in a neuron. They've chosen the B-cell system, which is actually quite a dangerous choice because B-cells are not a good place to live in the sense that they have naturally a short lifespan unless they are selected into a specific subset called memory B-cells. They are also subject to somatic hypermutation during memory cell selection. And that's why many of the lymphomas that arise in man are actually of B-cell origin, of post-germinal center B-cell origin, have gone through the somatic hypermutation process. So this is a virus that's taken a high-risk strategy, but has had to find ways to get over that. And the keys to its success are largely, if you think about the genes which EBV and the old world viruses have acquired, which are missing in the new world, what I should have said is, very interestingly, if you look at the seropositive rate among natural animals out in the wild, among the New World primates, the New World viruses are spread within their host population much less efficiently than the old. So maybe less than 50% of wild New World viruses will have their gamma-1 virus. So clearly transmission, the ability to be transmitted or the ability to establish latency has been improved with old world primate evolution, the acquisition of these genes. And so the genes that have been acquired by the old world viruses are largely to do with immune evasion during lytic cycle. Jianmin Zhuo and Andrew Hislop, who you heard, and Martin have done much to identify 4 or 5 such evasion genes, many of which are in this group, which have been picked up by the old world viruses. Efficient B-cell transformation is critical to establishing latency. But these viruses also have a key function that we simply don't understand, and Paul Murray has already alluded to this, is the ability to respond to physiologic signals during their life in the B-cell system. Okay, so why did I start to work on EBV, other than Tony Epstein gave me a job, is that I always think there's a kind of area of the map where virology, immunology, and oncology meet. And that's where I want to live, in that area. And whenever I've been working, and I've been lucky to get good people here, I try to keep that breadth so that we have different perspectives on the same agent. Okay. Now, of course, you can read everything you want in fields, but not everything you read is true. But on top of that, as I like to say to people when they start in that, read fields and then go and identify what they don't tell you, because that's what's important. I know that because I wrote the bloody chapters on EBV. Now, if you don't want to read Field's EBV chapter, of course, and it's quite good for pressing flowers, so, and drying conkers, actually. I find they are quite useful to keep in the office occasionally. But if you don't want to read that, you can always read this, which is the Poor Man's Field, recently published by OUP. You don't have to read it, actually, but it would be nice if you could buy it. And it came out about a month ago, and already someone at this, a guy from Singapore at the meeting last week said to me, "Oh yeah, I already got a second-hand copy on eBay." So that did wonders for my confidence. Okay, so I just want to try and, let's look across the landscape, and this is looking from 30,000 feet in a kind of William Withering-esque view, but occasionally descending a couple of times, just in honor of him, actually, to give you a little bit of data from two of the clinical research fellows who have recently been through the lab. So it is pretty much skimming the surface, and I hope that you'll forgive me if I do skim a bit too high. First of all, as Claire has already said, what I want to concentrate on is what we don't know, not what we do know. We know the virus comes in orally, and it can replicate in oral stratified epithelium. It's interesting, these are lesions on the tongue of immunocompromised AIDS patient, recognized by John Greenspan in the 1980s, actually, where he thought this lesion, which is a warty lesion on the side of the tongue, must be a papillomavirus. He started to look and was amazed to find that it was EBV. And there are stratified epithelium, the virus is replicating in a differentiation-dependent manner. It looks like a papillomavirus, Sally. And what's interesting is still we can't find any evidence of latent infection down here. So what this really means, we still don't understand. And we still don't know whether we can, with confidence, extrapolate from a highly immunocompromised AIDS patient to you or I, who are also shedding virus, but probably from microscopic replicative lesions in the oral epithelium. We don't know, actually, what happens in the oropharynx. Let me stress again, these are primate viruses. So we're hampered by the inability to really get an accessible animal model. We have rhesus, which if you can make a rhesus gamma-1 herpes virus-free colony, as Fred Wang has done, you can get experimental infection, and that's coming on stream now. But other than that, in a rather as yet poorly developed humanized mouse model, where you create the human immune system in an immunodeficient mouse and infect with EBV intraperitoneally, we have very little clue as to what the virus is doing in vivo, other than clinical observation and looking at samples. We don't know really what happens. We don't know which is the first cell to be infected. The virus can infect epithelial cells, as Claire told you, but very inefficiently. It can transfer from the surface of B cells into epithelial cells. Perhaps that is the entry route. And what's really amazing is that when the virus enters the B cell system, it's very quickly controlled, and viral loads go down rapidly, but The dirty little secret is when you look at people who've had a primary infection, had infectious mononucleosis, they go on shedding virus for months at quite significant levels. So whatever it is about the oropharynx, it's probably an immunoprivileged site, or there's some serious immune evasion mechanism that may relate to what Martin and Jan may have done, or may not. Things that we don't understand. And then the virus enters the B-cell system, and as Paul Murray had already told you this morning, actually, there is still a huge amount of debate about what actually happens here. But somehow the virus gets into a memory population, and we have to try and understand how these transitions work, because this is essentially a transforming function, and this is an antigenically silent infection. And how the downregulation operates and what are the physiologic signals that drive that are not understood. That's because we have focused too much on the in vitro system, where virus goes into B cells, drives proliferation, and is essentially an immortalizing infection. And this is through the coordinate expression of these 8 latent genes in essentially two transcriptional blocks. And one could talk forever about this, but I'm going to just leave it at that. So the co-expression of these antigens represents the fully transformed state as we see it in vitro. But when you look in vivo at the key tissue, which is the tonsil of infectious mononucleosis patients, not many infectious mononucleosis want to get bled, and very, very few want to get their tonsils out. There was a surgeon in Germany who was very strict about treating them in this way, and we had his telephone number for a while, and that was enormously helpful. And when you look, you've got the EBVs, which are short RNAs, non-coding, highly expressed, which are very sensitive markers of latent infection. Actually, their function is still controversial. EBNA2 and LMP1, but when you actually look at individual cells, you see that there are some expressing EBVA only, some with EBNA2, some with EBNA2 and LMP1, and some with LMP1 only, a little bit like what you see in Hodgkin's. So there is a multiplicity of forms that are co-resident within the tonsil, not just within the tonsil, but within individual B-cell clones. So Ralph Kuppers has microdissected these cells from the tonsil of IM and shown that these different latency forms exist within an individual immunoglobulin-marked memory clone. So we're missing things when we're working in vitro, and we need to work more on the influence of the environment on viral gene expression. Can we ever see the virus entering memory? I'm just fascinated by this. Does it really go through the germinal center or not? And here, actually, I had to say, although the germinal center idea is a seductive one and makes biologic sense, unfortunately, when you look in the tonsils of mononucleosis patients, all of the infected cells are outside the germinal center. The transitions are happening in an extrafollicular area. So this transition is something we're still trying to get at, and we had an idea, and I'll just come down from 20,000 feet here now and just show you some data from David Burns. We've been looking at what happens when a hemopoietic stem cell transplant patient who's been devoid of B cells has given new stem cells and then generates a new lymphocyte system. What happens to virus? Because these individuals are very prone to post-transplant lymphoproliferative disease. They're basically immunocompromised. Here is a normal person. We're looking at their B-cell population using IgD and CD27 as markers. The blue cells are memory cells, are non-switch memory cells. The purple cells are actually conventional memory, and the green cells are naive cells. And when you look at the green naive population, you can see in healthy, normal people, there are a few cells with a CD38, CD24 marker, which is a transitional cell. These are cells which have just come out of the bone marrow and are becoming naive. Then we go to a transplant patient, post-stem cell transplant patient, who has not reactivated EBV. He's got virtually undetectable EBV load. His B cells look quite different. Most of them are naive. There's just a few memory cells here, 1.3%. And most of those naive cells are actually transitional. This is exactly what you'd expect from a repopulating B cell system. But there are some patients who reactivate EBV to very high levels. 2-3 months after transplant, these are at very high risk of lymphoproliferative disease. They're hit with rituximab to try and control that these days. But we get them just before they're hit. So here's an example. All of a sudden, we've got 150,000 genomes per million B cells in this, a huge load. And look at the distribution of the B cell subset in these individuals. They've got naive cells, but most of the B cells, 50% of his B cells, are memory cells. The naive have got lots of transitional cells, so suddenly this population has arisen and EBV has reactivated. Are they the same cells? And when we look now, we can sort those memory B cells. And David does this and does the load and finds that essentially all of the virus is there. This is the total white cells. Here are the memory B cells. All of the virus is in that new memory B cell population, which has emerged from a repopulating stem cell transplant patient. And more to the point, actually, when we look at virus load in these cells, there is no replication going on. Every one of these cells has got between about 5 and 30 episomes, which is the classic marker of a virally transformed B cell. And many of these cells actually, though I've missed the slide now, show markers of transformation. They're Ki67 positive, and they look like they're expressing transforming genes. So the virus is driving, through a transformation event, the repopulation of memory B cells in the stem cell transplant patient. Patients who do not have this have essentially no memory B cells yet. They're basically all naive and transitional. So somewhere in the lymphoid tissues, there is an event going on. Either the virus is finding memory B cells and transforming them selectively, or is infecting naive cells and driving them into a memory state, changing their phenotype, possibly changing their genotype. And we need to get back into lymphoid tissues to see this happening. Okay, there are many other questions. I won't pursue it, but, you know, what controls this? What's the physiologic signal that says, okay, episome, just sit there, attach yourself to the human chromosome, and don't only express EBA RNAs. Don't express antigen. And then what are the triggers that reactivate? Because remember, this is a latent reservoir which has no evolutionary advantage to the virus unless the virus can get out and escape, get back to a secondary pharyngeal epithelium and start to shed again. Okay, we don't know really what controls those events, although there are many theories. Okay, so this is the classical virology, if you like. Let's look at the virus as an immunogenic target or as an immunopathologic agent, as it turns out. So here we have the transformation experiment in vitro that we were doing way back in the early 70s. And then towards the end of the 70s, as T-cell markers, I mean, it's hard to believe now, but we didn't really know what B and T cells were until about 1972. Then it was shown very nicely in George Klein's lab that these were B cells being transformed. When people looked at the atypical mononuclear cells that we were in mononucleosis, and we already knew this was an EBV-driven disease, everyone said they're transformed B cells, but they weren't. They were reactive T cells, largely CD8. It was that point where I really thought, I better learn some immunology. And I went to Brisbane to have a year in the sun with my friend Dennis Moss, who's wearing this orange jumper, which his wife knitted after the birth of their first child, and he's still got the jumper, and it's particularly holy now, and I didn't get the picture of it, Martin, but he was wearing it last week for special request. And what Dennis and I did was, I arrived there by accident thinking, I'll play some cricket, have a nice year, and we worked day. Because we came upon something that we hadn't seen before. Or Dennis had seen it and then had passed on to other things. And I rediscovered it when I went to his lab. And for the first time, instead of using cord blood as our target cell, I used B cells from our cells. And I kept saying to Dennis, look, Dennis, I'm getting transformation, but then my cultures are dying. There's something wrong with your medium here. He was not impressed by that, and he said, look, and he took me to a paper that he published in 1975 that I'd forgotten to read, which described this phenomenon. And I said, why aren't you working on it? He said, well, I got sidetracked. And immediately I went around the lab and looked for the serologic status of our donors. And within 3 months, we were clear that this was a phenomenon that was only seen in virus-immune people who had the virus. And it was clearly T-cell mediated, we knew that. And it took a while working in Bristol, here, and of course Dennis working too. We worked in tandem on this. It's a phenomenon principally mediated by CD8 T cells, but also CD4 T cells are there. These are classically restricted through class 1 and class 2. When we first discovered this, we discovered that we were working on MHC restriction in man. Andrew McMichael, my great friend in Oxford, is working on influenza. We're the only two groups who were finding restriction. And this was quite important because the restriction phenomenon, which had been described by Ralph Doherty and Ralph Zinkernagel and Peter Doherty in mice, there was some debate still about its relevance to man. But we managed to show that this was the case. We later found that what the antigens were and then what the peptides were. So this took us on maybe 15 years or so, but then we had another stroke of luck, which was I met Paul Moss. Paul Moss now works here, and this is a contemporary picture. In fact, it was taken about 2 hours ago. Paul, working in HIV in Andrew McMichael's lab with Mark Davis as the developer, had shown the usefulness of a technology whereby we could visualize T cells with particular receptors specific for particular combinations of class 1 molecule and peptide. And we had our viral peptides, and we knew our class 1 molecules. So we could build these reagents. So here's a picture, a cartoon of the T cell surface with the T cell receptor here. Here is the virus-infected cell with a viral peptide in a class 1 molecule. And what, of course, Mark Davis had done was to generate these reagents, which were basically tetrameric versions of the peptide HLA complex, labeled with phycoerythrin, so you could put them onto T cell preps and say, is there anything there that sees my peptide? And we started to do this with infectious mononucleosis, because I should have said that in infectious mononucleosis, the total CD8 T cell pool is 5 or 10 times larger than in healthy people. And that is coincident with the disease. And the cells are largely activated, as I showed you on a previous slide. And when we put the tetramers on, we could hardly believe our eyes. Because here, this is an A2 positive patient. And these are 3 peptides which come from EBV proteins. We just show the first 3 amino acids as a shorthand, which are presented in the context of HLA-A2. And the tetramer is picking up 33%, 12%, and 1% of these T cells in this highly activated patient. If we go way down the track here, this is a long time, they've all gone back to a kind of low-level memory phase. We had to pick these cells out and clone them and look at their receptors and look at their specificity. Otherwise, we couldn't believe that the response was so large. So essentially, nearly all, or the majority of this highly activated elevation of CD8 T cell numbers is driven by the virus. And it's the most extreme example of a virus-driven T cell response in primary infection. You see it very occasionally in HIV, but the sort of pale reflection in most other viruses. You'll see small responses, again of activated cells, which are specific. Something has gone wrong in mononucleosis. It seems to generate this kind of expansion, which is exactly coincident with the disease symptoms. So actually, this looks like an immunopathologic disease. We know that cytokine storm derived from activated T cells release pro-inflammatory cytokines. And in other circumstances, you get severe febrile illness from that. And this is what's happening, actually. So written now, superimposed on the diagram of the viral infection is the immune response. And most of this highly activated response is derived against the lytic proteins, actually, and the latent proteins follow on. And then this dies away. And so we knew what was happening, but many questions still remain. I mean, most importantly, how is it that some people get mononucleosis? Other people get infected even in adolescence and do so without obvious symptoms. And children pick up the virus regularly, but hardly ever develop symptoms. We wanted to get at that. And there's a bigger question, which is, how do the events here relate to the virus load long term? How is the position of equilibrium set? Because in this room, I can guarantee that there is a 10,000-fold difference in the level of EBV that we carry. So everyone has got their own idiosyncratic level, and they carry that for life unless they get immune suppression. We don't understand at all how that variance is generated. Now, something about then asymptomatic infection. What distinguishes IM from asymptomatic infection? Here's the second parachuting down just to show a result, and this is results which, if I had the right slides with me, it would have Shami Jayasuriya's name on, and the MRC Gambia labs where she worked, supervised by Andrew Hislop, who you've heard from us. And we logged on to a vaccine trial where infants between about 9 and 18 months were being vaccinated and bled before vaccination and after. And they had spare peripheral blood lymphocytes, and we looked at them. And we were able to identify 5 people who serologically, 5 infants, who were having primary infection at the time by EBV IgM. Their viral loads were high, but look, the viral loads in all the children were high. This is acute IM patients. These are Birmingham University medical students, God bless them, who have donated their blood in, threatened that we'd fail them if they didn't give blood. Yes. So virus load is high, but the infection is apparently asymptomatic. What about the T-cell response? And here we can look at it now. We're using the same tetramers now. And we're looking and look, in this child here, on day 1, high load, 15% of their CD8 T cells are specific for one viral epitope. 2 weeks later, it's already going down. And then 14 months down, it's down to 1 or 2%. This is a classic picture that you might see in infectious mononucleosis in terms of the quality of the response and the activation phenotype of the response. But what's different about these is, look here. Here are the 6 primary infections with that response going on. Total CD8 T cell numbers have not moved. And so there is no elevated lymphocyte count. There's no overall CD8 T cell expansion. Virus load is high, and there is a real EBV-specific response going on, but it's not magnified. So what distinguishes a symptomatic from a non-symptomatic infection is apparently not virus load per se, but something to do with the control over CD8 T cell homeostasis that we still don't fully understand. So IM is an immunopathologic disease, and we think that the symptoms, which are febrile symptoms, are largely driven by overexpansion of the CD8 T cell response, and there is some NK cell activation. Pro-inflammatory cytokines are sky high in the plasma, many of them. There is a fatal form of IM. I haven't got time to tell you about it, but I just put it in because there's a huge amount of data coming in now from new primary immunodeficiencies. XLP is the most famous, X-linked lymphoproliferative disease, which actually affects the SAP gene, which encodes a protein which is on the communication link between T and B cells, and between NK and B cells. And if you have SAP gene mutation, then T cells and NK cells cannot see B cells. And that is why these people are peculiarly sensitive to EBV infection and not to measles or to CMV or to influenza or to whatever, because it's a BT-specific defect. Work that was done originally by Antonio Marretta for NK cells and by Andrew Hislop for XLP. And there is this syndrome which Claire told you about, virus-associated hemophagocytosis, which is seen commonly not commonly, but is seen and relatively well known in pediatric cohorts, particularly in Southeast Asia. We're working with a Vietnamese hospital, because what is happening there is also a T cell and NK cell expansion, and also cytokine storm, but there the cytokine storm is coming from virus-infected T cells, virus-infected NK cells. So same endpoint, but a different mechanism, route to it. And there may be more autoimmune diseases. And one version of this talk, which unfortunately I left at home last night, I was bold enough to say 2 words: multiple sclerosis. I'm going to stop and we'll discuss it in the bar. But that is another fascinating new aspect of EBV work. Okay, so I've talked about that. Now, what about EBV as an oncogenic agent? Just to complete the triad. Martin, tell me when I'm finished. As Paul Murray has told you, I'm very grateful for Paul for giving that elegant introduction. I can skip these. Post-transplant lymphoproliferative disease is a virus-positive lymphoma, but so are these other malignancies at different levels of EBV association. And as Paul said, the diffuse large B-cell lymphomas are largely B-cell lymphomas of the elderly, or B-cell lymphomas in association with artificial pneumothorax, which is a source of chronic inflammation in the lung. There are these lung B-cell lymphomas, DLBCL-like, that are EBV positive, that are arising through chronic inflammation. And several non-B-cell tumors, and we've talked about these to some extent. Let me just say something about gastric carcinoma, because it's about 8% actually worldwide of gastric carcinomas are EBV positive in every malignant cell. Is this just a chance spurious effect? It looks like no. It looks like it's specific because the actual, first of all, the epigenotype of these EBV positive tumors is quite distinct. They're highly methylated, whereas conventional adenocarcinomas, EBV negative, the EBV negative subset, the majority of gastric adenocarcinomas, linked to Helicobacter pylori infection, have different epigenotypes. And there are now specific mutations coming out, and there's a paper coming in Nature soon, where there are some P3I kinase mutations which are specific to the EBV-associated form. So this looks like a pathogenetically distinct subset of gastric carcinomas. Now we really start to take it seriously. It's not a casual infection. But I just want to say something about Burkitt lymphoma. Why? Because historically, it's the first EBV-associated cancer. And actually, I'm rather pleased, actually. We went back to Tony Epstein's, at his instigate, went back to Tony Epstein's original strategy to get new biopsies. And in the early 80s, actually, and right throughout the 90s, it was possible to get the stuff. And here is Ching Wen Yao's Gang of Four. Here's Martin, in case he's looking a bit slimmer than he does there. Here's Cleo, who went on to cure post-transplant lymphoma with T cells, very famous. We came to Birmingham, and they were very good to us, because I've been through a succession of deans and heads of college and this rubbish that goes on in universities. And they all say, Alan, just go away. We know you're no good at meetings. Just get back to the lab and do something useful. So I was extremely grateful that Birmingham did this for us, because in the end, it allowed us to really get some work done on what we were good at. Martin first recognized, when we started to make these cell lines, that they generated heterogeneity in culture. Some lines remained single cells and retained a classic Burkitt phenotype. Other lines progressed and became much more like in vitro transformed cells, changed their phenotype, and in fact very much looked like in vitro transformed lines, except they still carried the MYC translocation. When we looked more carefully at this, we still don't understand why some tumors do this and others don't, actually. But the sort of eureka moment, if you like, is when we started doing blotting. And this is one of Martin's blots. I know it's Martin's because I'd never get anything so beautiful, where the LCL-like cells are expressing the full spectrum of latent proteins. And this is EBNA1. So we knew that from that point, we knew that we'd been looking at the wrong form of latency in our in vitro models. We knew that there were other forms. And look at this. MYC is high in these cells, which are the original Burkitt phenotype. Cell lines which have drifted to become LCL-like have actually downregulated the translocated MYC. Really fascinating, something that's still not been actually explained properly. And this, we worked with Elliot Keefe, and we actually got the transcriptional program in these cells, and of course EBNA1 was being expressed from a completely different housekeeping promoter, which is not activated in the conventional transforming type of infection. And this opened the floodgates now because we had two tumors that had different forms of latency, and it didn't take that long to go through everything else and say, okay, we've got a whole spectrum here from EBNA1 only in Burkitt's through EBNA1 and the high levels of the LMPs in Hodgkin lymphoma, and in the non-B-cell tumors, largely EBNA1, and then variable levels of the LMPs, a kind of latency 1/2 halfway house. And this, again, reflected differential latency programs. So everything was determined transcriptionally, essentially, in these different types of infection. And I just want to spend one moment saying, well, I mean, we knew about this for the past, how long? 30 years. That's embarrassing to say that. Not quite, 25 years. And we're still working on the implications of this. As I've said, these different programs reflect different patterns of latent promoter usage. And those patterns are likely determined by the identity of the infected cell. We know that the infected cell transcription factors can determine what is expressed and what is not. Obviously, the viral episome then is then the whole architecture of the episome is determined by the types of methyltransferases, etc., which are in the infected cell. So this is a way in which the virus has developed flexibility. And I think, obviously, these forms of latency have not evolved to help tumorigenesis. That's a nonsense. They have evolved to give the virus flexibility as it lives in the B-cell system. And how that works at the molecular level in different cell types at different stages of B-cell differentiation is something that we still are working on, and like many others, and we're still only partway there. And why are they there in tumors? Well, so I think many of these alternative forms of latency are transiently expressed in vivo when the cell is going through a particular phase of B-cell differentiation. But they've been captured in the tumors, and the reason they've been captured is that they contribute to the malignant phenotype. When the virus is there, and there is a step in the oncogenic process that can be facilitated by that form of latency, and it happened to be present at in that cell at that time, it will be captured because it gives the infected cell an advantage, probably a survival advantage rather than a growth advantage. So apart from post-transplant lymphoproliferative disease, which is largely driven by the virus, these other viral tumor associations reflect different forms of viral contribution, largely, we believe, to do with survival from apoptosis rather than growth per se. So we're still working on when and how these viruses act during tumor development. And then, of course, we have to think, what can we do about it? obviously we have a responsibility to think about what this means in terms of tumor therapy or in terms of tumor prevention. And I've taken out a whole lot of slides, you'll be pleased to know, which referred to Graham Taylor's work, where we've actually, so if we just go back for a moment, here. Now what I wanted to say is that these two tumors now, every case of nasopharyngeal carcinoma, and about 8% of all gastric carcinomas, including a small subset of gastric carcinomas which have the same histology as NPC, with lots of inflammatory cells, these constitute about 170,000 new cases of cancer per year, globally. That's approaching 1% of the total cancer burden. They far outweigh all of these tumors, all of these other tumors actually. They far outweigh numerically. So if we're really thinking about therapies, we need to think about how can we get at these patterns of gene expression, or how can we use these patterns of gene as targets for immune therapy, or how could we look at how LMP2 operates and how could we drug it? We've done something along those lines. We've developed a therapeutic vaccine which is now being used, and we've been through the first clinical trials in Hong Kong, the paper's published, where we're looking at the effectiveness of a vaccine, a vaccinia-based vaccine expressing EBNA1 and LMP2 as a fusion protein to reactivate EBV, LMP, and EBNA1 specific CD4 and CD8 responses. We get good responses. All of the work was done in NPC patients who were disease-free, and we got responses in 15 out of 18 patients treated in Hong Kong that way. Immunologic responses, not clinical responses, immunologic responses. And the size of the response equated with vaccine dose. I should have put the slides in because it's a classical William Withering experiment, actually, where we now know the best dose to give the patients, and we're now entering a second trial where we're giving actually 6 successive doses of this MVA-based EBV vaccine as a therapeutic option to patients who have developed NPC, who've been treated by conventional radiochemotherapy and are now at high risk of relapse as determined by levels of EBV DNA in the plasma. You can actually use EBV DNA in the plasma as a biomarker of tumor mass. So we're very interested to know whether we can affect the long-term remission or relapse rate among patients with this strategy. The key thing is to get immunologic responses that are retained rather than just transient. It's one small step in a much longer road, actually. But as we're at 50 years and we're thinking about the future, here are the numbers, and this is the quote that will give you those numbers, and just to say how important these tumors are. But thinking ahead now, I have to go back to a conversation I had with Tony Epstein in 1976, saying, we've got a virus, we've got cancer, let's just vaccinate and prevent infection. And he had a monkey model in those days. It was pretty crude, but he did prevent lymphomagenesis. He didn't prevent infection by reducing the dose of virus with a neutralizing antibody response. So the questions that the field faces now for the next 20 years, I believe, is can we provide a prophylactic vaccine that provides sterile immunity? There are many questions we don't know. I must say, within the field as a whole, there's extreme skepticism over this. And one of the reasons is that if we look at you or I, who've been carrying the virus for maybe 20 years, now that we've got deep sequencing, we can find 2 or 3 viral strains in many people. So the question is, did you pick up strain 1 from the first girlfriend, strain 2 from the second, strain 3 from the third, or did you pick up all 3 strains from the first one? And we're trying to do those sociologic experiments. But in the meantime, But the fact that people had multiple strains saying, oh, forget it, you know. Even if you get one virus, somehow you're not immune to reinfection. Then we went to mononucleosis patients, where we had been isolating EBV strains from mononucleosis patients in vitro by transformation. And I had 20 patients. Qingwen Yao made 20 cell lines from 20 patients from the blood and 20 cell lines from the same 20 patients from the throat. And he got the same strain. All 40 isolates from one patient were the same. And that happened 20 times. So everyone picks up one distinct viral strain until you go into the blood and the throat, and then you amplify, you do deep sequencing, and you find that there are more strains. So even a primary infection will give you more than one virus. Something that doesn't happen in the HIV field, I think, Robin. One pass on. So I lost a lot of money, actually, on that bet, that we'd only pick up one strain in primary infection. So we still don't know whether the infected host can acquire more strains. If we make a prophylactic vaccine, do we have to do better than nature and produce sterile immunity? Would we want to produce sterile immunity in young children if they were going to come down later with infectious mononucleosis in adolescence? Another point to debate. And also, the protagonists of the vaccine currently are dominated by the idea that you'll never get sterile immunity, but what you will do is reduce the virus load that a new person acquires by developing neutralizing antibodies. And my question is, would reducing the virus load, the initial virus load, make any difference to the long-term load? And because we have no idea how the long-term load is set, we don't know how the virus set point is established. We don't know that reducing the virus load will reduce the long-term load. There is some evidence from monkeys that that might be the case, but we have nothing in man. So there's a lot to do. How would we design such a vaccine? Everything is based on the major glycoprotein at the moment, GP340, but that glycoprotein gets the virus into B cells. It's not the viral envelope protein that gets them into epithelial cells. So could it be that we need neutralizing antibodies also against epithelial cell infection, however that works, through the other glycoprotein, so-called GHGL complex, which is responsible, part of the mechanism responsible for epithelial entry. Which glycoproteins, and would we get T-cell responses to be protective? We probably need T-cell responses in reserve to help, and then which are the most important antigens to target if we're going to immunize the T-cell system and make that a complementary approach to neutralizing antibodies? We don't know. Okay, enough questions for one day. Yeah, it's time for drinks. Here is Martin. I'm trying to give him a bear hug, but it's actually quite difficult to give Martin a bear hug. But this was at the EBV meeting, and you can see that we're feeling quite happy with ourselves that actually we haven't solved that many problems at all, and there's so much still to do. And that's what science is about, ever-expanding horizons. So, you know, as you look at it, you think, well, we solve one problem and we make three more. So this is this is the life of a scientist, and that's why it's a good thing. We've been at it for, Martin, 30 years. I've been at it for 40 years. Here we were in 1964. Actually, this is my grandson, Will Fitzsimmons. He looks remarkably like me at the age of 5, which is slightly worrying. So I'll try and talk him into a career in EBV research. Thank you for your attention.